Bismillahirrahmanirrahim. Assalamu alaikum. This is Dr. Hooria Aamir, and I welcome all the students of second year MBBS to today's lecture regarding the gastrointestinal physiology. Although this topic is lengthy but quite interesting, and I hope all of you will do it regularly with me so that the subject will be easy for you. So, let's talk about what is GIT physiology and um, what actually are the learning objectives that after the end of the today's lecture you should be knowing about the general principles of gastrointestinal function, the motility, the nervous control, the blood circulation, how the gastrointestinal smooth muscle is functioning as a syncytium. And what is that? That is the one unit of muscles. The electrical activity of gastrointestinal smooth muscle. Action potential, changes in voltage of resting membrane potential, calcium ions, and muscle contraction. Tonic contraction of some gastrointestinal smooth muscle. Now, what are the general principles of gastrointestinal function, motility, nervous control, and blood circulation? The alimentary tract, which is providing the body with a continual water supply, electrolytes, vitamins, and nutrients. And to achieve these requirements, what is needed is that the movement of food should be there throughout the alimentary tract. There should be secretion of digestive juices and the digestion of food is needed. Then absorption of water, various electrolytes, various nutrients, vitamins, digestive products should take place. Then circulation of blood via GIT organs is needed to carry away the absorbed substances to far-flung areas of the body. Then control of all these functions by the local nervous and hormonal systems so that the maintenance of homeostasis of the body is done. So the all principles of GIT should be there, and these all are actually needed in harmony. There should be a harmony with that of the blood circulation, with nervous control, with the movement of GIT, so that the proper movement of food should take place, proper secretion of digestive juices, digestion of food, absorption of food, circulation of blood via whole GI tract, then control of all functions are there to maintain the homeostasis. Now look at this diagram. You see the whole digestive tract, the whole gastrointestinal tract starting from mouth till that of the anus. And this is actually the tract in which there is ingestion, digestion, secretion, absorption, and excretion. They all are taking place. Now, general principles of gastrointestinal motility. Now, physiological anatomy of gastrointestinal wall is that the layers of gut from outer surface towards the inwards, they are that first of all, excuse me, there is serosa outside, then there is longitudinal smooth muscle layer, then there is circular smooth muscle layer, there is submucosa, and finally inside there is mucosa. So always remember that there is presence of 5 layers in the gut wall, which are starting from outside to inside. We have serosa, two muscular layer of smooth muscles, then we have submucosa and mucosa. See, 5 layers starting from outside to inside. There is serosa, then there is longitudinal muscle layer, then there is circular muscle layer, there is submucosa, and finally there is mucosa. Now, always remember that the gut, the gastrointestinal smooth muscle, functions as a syncytium, as a one motor unit. So, individual smooth muscle fibers in the gastrointestinal tract, they are 200 to 500 micrometers in length. They are 2 to 10 micrometers in diameter, and they are arranged in bundles of as many as 1,000 parallel fibers. In the longitudinal muscle layer, the bundles extend longitudinally down the intestinal tract, whereas in the circular muscle layer, they extend around the gut. The muscle fibers, they are electrically connected with one another via a large number of gap junctions, which allow low resistance movements of ions from one muscle cell to the next. Each bundle of smooth muscle fibers is separated from the next by loose connective tissue, but muscle bundles fuse with one another at many points. Therefore, each muscle layer functions as a syncytium. That is, when an action potential is elicited anywhere within the muscle mass, it generally travels in all directions in the muscle. Now, we should be knowing about the electrical activity of the gut, electrical activity of GIT smooth muscle. Now, the smooth muscle of the GIT is excited by almost continual slow intrinsic electrical activity along the membranes of the muscle fibers. This activity has two basic types of electrical waves, which are slow waves, spikes. The voltage of the resting membrane potential of the gastrointestinal smooth muscle can be made to change to different levels, and this can have important effects in controlling motor activity of the gastrointestinal tract. See how the spikes they are there, what is depolarization, what are the stimuli which are like stretch, acetylcholine, parasympathetics, slow waves, resting membrane potential, C, which is around minus 50 millivolts. Slow waves. Most of the gastrointestinal contractions they occur rhythmically, and this rhythm is determined mainly by the frequency of so-called slow waves of smooth muscle membrane potential. Their intensity usually varies between 5 and 15 millivolts, and their frequency ranges in different parts of the human gastrointestinal tract from 3 to 12 minutes per minute, about 3 in the body of stomach, as much as 12 in duodenum, and about 8 to 9 in the terminal ileum. The rhythm of contraction of the body of the stomach is usually about 3 per minute of the duodenum, about 12 per minute, and of ileum, about 8 to 9 per minute. So, the cause of slow waves is caused by complex interactions among the smooth muscle cells and specialized cells called interstitial cells of Cajal, and they are believed to act as electrical pacemakers for smooth muscle cells. These interstitial cells they form a network with one another, and they are interposed between the smooth muscle layers with synaptic-like contacts to smooth muscle cells. The interstitial cells of Cajal undergo cyclic changes in membrane potential due to unique ion channels that periodically open and produce inward pacemaker currents that may generate slow wave activity. Spike potentials. The spike potentials, they are true action potentials. They occur automatically when the resting membrane potential of the gastrointestinal smooth muscle becomes more positive than about minus 40 millivolts. The normal resting membrane potential in the smooth muscle fibers of the gut is about minus 50 and minus 60 millivolts. So there is just 10 millivolts. The muscles, they have to go towards positivity to reach the threshold level for automatic uh, potential development. See how it occurs. Minus 50 to minus 60 millivolts resting membrane potential and just minus 40 millivolts, that is actually the threshold, and then the depolarization is going to take place automatically. Now the peaks of slow waves temporarily become more positive than minus 40 millivolts, and the spike potentials, they appear on these peaks. The higher the slow wave potential rises, the greater the frequency of spike potentials, ranging between 1 and 10 spikes per second. The spike potentials, they last 10 to 40 times as long in GI muscles as the action potentials in large nerve fibers. Each GI spike lasting as long as 10 to 20 milliseconds. Now, action potential. In nerve fibers, the action potentials, they are caused almost entirely by the rapid entry of sodium ions through the sodium channels to the interior of the fibers. Whereas in these gastrointestinal smooth muscle fibers, the channels, they are responsible for action potentials, they are quite different. And they allow especially the large numbers of calcium ions to enter along with smaller numbers of sodium ions, and therefore are called calcium-sodium channels. The slowness of opening and closing of the calcium-sodium channels accounts for the long duration of the action potentials. The movement of large amounts of calcium ions to the interior of the muscle fiber during the action potential plays a role in causing the intestinal muscle fibers to contract. Then we should be knowing what are the changes in voltage of the resting membrane potential. The baseline voltage level of smooth muscle resting membrane potential can also change. Although under normal conditions the resting membrane potential, that is between minus 50 to minus 60 millivolts, which is on an average minus 56 millivolts, but multiple factors, they can change this level. When potential becomes less negative, which is called depolarization of the membrane. When the potential becomes more negative, it is known as hyperpolarization. See, what is this hyperpolarization? That is more towards negativity, and depolarization, that is towards positivity. Now, factors which can depolarize the membrane, that is, can make it more excitable, they are like stretching of the muscle. Then, stimulation by acetylcholine released from the endings of parasympathetic nerves. Then, stimulation by several specific gastrointestinal hormones. Then we can have the factors which can make the membrane potential more negative, that is hyperpolarize the membrane and make the muscle fibers less excitable. And what are they? They are epinephrine or norepinephrine on the fiber membrane, stimulation of sympathetic nerves which secrete mainly norepinephrine at their endings, entry of calcium ions, they cause Smooth muscle contraction. Smooth muscle contraction occurs in response to entry of calcium ions into the muscle fiber. Calcium ions acting through a calmodulin control mechanism activate the myosin filaments in the fiber, causing attractive forces to develop between the myosin filaments and the actin filaments, thereby causing the muscle to contract. The slow waves, they do not cause calcium ions to enter the smooth muscle fiber, only sodium ions. The slow waves by themselves usually cause no muscle contraction. It is during the spike potentials generated at the peaks of slow waves that significant quantities of calcium ions do enter the fibers and they cause most of the contraction. Tonic contraction of some gastrointestinal smooth muscle. Some smooth muscle of gut exhibits tonic contraction as well as or instead of rhythmical contractions. Tonic contraction is continuous and is not associated with the basic electrical rhythm of the slow waves but often lasting several minutes or even hours. Causes of tonic contractions. The causes can be the tonic contraction is sometimes caused by continuous repetitive spike tensions. Tonic contractions can be caused by hormones or by other factors. A third cause of tonic contraction is continuous entry of calcium ions into the interior of the cell. So, this is just the introductory lecture regarding the gastrointestinal tract physiology, and I hope you'll all do it regularly uh, from GIT so that the topic should be easy for you to prepare. So, till then, Allah Hafiz and stay blessed.